I'm MC Toon, the worst rapper in the world, and I've got with me Dr. Wilson from Debunk the Funk with Dr. Wilson. How are you doing, Dr. Wilson?
Hey, Mike, I'm doing good. How are you?
I am great. I've been looking forward to this. I am a big fan of your channel. I hope you find the link in the description to his channel. Go subscribe. It's excellent, excellent information. You do a video about every Tuesday. Is that right?
Yeah, I try to do every Tuesday. Yeah.
Did you have one today?
I did, actually.
Okay. I was a bit busy preparing for this and had some other things, so I missed it. So I'll have to catch it later. So everybody catch it. What was the topic of the one today?
So it's a topic that is close to my heart, actually. It was RFK Jr., if you've heard of him.
Oh, yeah.
Very famous anti-vaxxer, son of Robert F. Kennedy. And yeah, he has said a lot of anti-vax stuff over the years and has put a lot of money towards promoting the anti-vaccine movement. And so I decided to finally debunk him. And if you're interested, there's a link in the description of that video to donate to the Autistic Self-Advocacy Network, which is an advocacy group that's run by autistic people, which I thought was appropriate since anti-vaxxers just always kind of reduce autistic people to a disability. And I think that's kind of gross. So
yeah. So well, good. That's pretty cool. All right. I'm going to show here. So the point of this particular stream is addressing some claims by a guy named Logan Mannix, who commented on your video that came out last week. And he's been making lots and lots of claims, and we're going to look at them. So if people want to see the whole interaction, there is one thread on that video that's got nearly 200 comments already.
Oh, wow. That's interesting, because I usually don't read through all my comments anymore because it would take too much time.
Yeah, and that particular one is a lot. And so I've been there's a guy in there, Colin, who thank you very much. We've been talking back and forth a little bit, and it's been interesting that this Logan Mannix guy, who issues challenges to debate, as you see on the screen right now, won't show up for a debate. So there's two parts to his claims. Some are about things that are biology and medicine, which would be Dr. Wilson's strength, and some things about 5G and some other technology, which I can address. So let me read this here. Said COVID-19 he says doesn't exist, and there's no virus at all. I will challenge and debate anyone on the subject, including medical experts. You will lose. Be scared. He won't, and in fact, he won't even show up is what'll happen. What COVID-19 is and what it has been getting people sick is 5G. So he says that of course, he is completely and totally illiterate in all of these topics. So let me see here. Oh, this one is particularly bad. This is getting, now I have sent him a link, and he has obviously not accepted it. So if people in the chat, if he does show up, he has the link. He should come on, give him the Vicky treatment. You know what that means, people. So here is his claim. If you remember that nurse that got the shot and then passed out, he says people, let's see, put her video next to the video, people falling over the streets. They're identical. It's a load of crap. Oh, maybe this is the wrong one. Oh, the nurse. She's right there in the first. People, despite what the media says about the nurse who collapsed, the truth is she is now dead. Of course, she came on afterwards and said, I'm not dead.
Yeah,
she did not just faint. The woman died. Of course, he has no evidence of this other than just seeing the video. And it's a common thing of the everything deniers that yeah. So, all right, let me find, maybe you want to address a little bit on the evidence that would be necessary to determine if a virus is real and if specifically how COVID-19 may or may not be determined to be real.
Yeah. So It's actually pretty simple now that we know what viruses are. It's not that hard to demonstrate that one, they exist, and that two, they cause disease. So obviously the challenge back 100 years ago was that viruses are really tiny. So if you think of how small a human cell is, a virus, most viruses are about 1,000 or 100 times smaller than that, which is still an order of magnitude smaller than bacteria. So with such tiny things, it's kind of hard to detect that they're real without understanding what they are or having certain technology. And another caveat that I think that a lot of COVID deniers kind of really miss is that viruses need a host cell to grow in, right? They're this unique thing where they're not quite a parasite because they don't have any metabolic function outside of a cell, outside of a host. But once they enter a host, then that's the only time things start happening. So they absolutely need hosts. So what to do if you're trying to figure out if a virus is real, you just take a sample from a patient. You think that they're sick with a virus. You swab their throat, take some fluid from their lungs, whatever. You take a little bit of that fluid and you put it into a dish, a cell culture dish, where you're growing cells that the virus could infect. And then the virus infects those cells. And lots of COVID deniers have a problem with this. They say that's not a pure culture. You're not really isolating the virus. You're not purifying the virus. But we'll get there in this conversation, I'm sure. But that's what you do. You infect a culture of cells with this fluid, and then you can watch the cells to see if they get sick in that dish. You can actually watch them die in real time. And then once a lot of the cells have been infected and have helped produce more virus, then you can take just the liquid, just the fluid off of those cells. No cells come with that fluid. And then you have your virus, you have your isolate. And then you can sequence that virus, you can get its genome, you can study the code that codes for all the proteins that the virus needs in order to be a virus. Or you can do other things with that isolate. So that's really all you need to do in order to obtain an isolate. But in order to demonstrate that the virus causes disease, you would have to satisfy what's called Koch's postulates. And you might have heard some COVID deniers talking about this, I assume.
Yes.
Yeah. So the confusing thing, I think, for COVID deniers is that Koch's postulates is slightly different when it comes to viruses compared to bacteria.
So yeah, Rivers is the guy that developed that.
Exactly. Yeah, Rivers. Rivers modified Koch's postulates, and nowadays there are 6 requirements. You have to isolate the virus. That's the first part of what I described. You have to cultivate it in host cells, which is also part of what I just described. You have to demonstrate that it can replicate in cells. You have to demonstrate that it's filterable. So there are certain filters that when you pass liquid through them, it'll catch all the bacteria, all the human cells, all the fungi, everything else, but it'll allow the viruses to come through the filter. So when you pass the liquid through the filter and you collect what comes through, and that liquid is still infectious, it's still causing your cells to get sick in the dish, that's demonstrating filterability. Then once demonstrate that, you need to demonstrate that it causes disease in either the original host species. So if it's humans, you can't really experiment on that. That's not ethical. So you move to a mouse model or another animal model. So demonstrate that the virus can cause disease in an animal model that is similar to the original animal that it caused disease in this case, humans. Then you can re-isolate the virus from that animal. So again, collect a sample from that animal who got sick after you infected it, and then re-isolate the virus by taking that sample, putting it back into a cell culture. And you also, the final requirement is detecting a specific immune response to that virus in the host organism. That means the organism formed antibodies against the virus, or it had some sort of cellular immune response to it, something like that. So those are the things we look for when trying to demonstrate that a disease is in fact caused by a virus and not a bacteria or a fungus or 5G.
Or an exome, as he sometimes
the exome. Yeah, that's a weird one that popped up in the COVID era.
Is that a new thing, or is it just they dug up something that's been kicking around in some, disproven dumb thing for a while ago?
It was new to me during the COVID pandemic, the idea that exosomes are viruses or exosomes or other way around. It was weird for sure. I don't remember when I was reading about it whether or not I found if that idea has been around for a while, but either way, it was just like, why would anybody think that? I mean, there are whole fields of study dedicated to studying exosomes. We know pretty well what they are. They're definitely not viruses. They're very different.
So yeah, let me read. It's kind of wordy. I'll try to find the part that's quick here. We're up on screen as one of his comments. It says your body secretes exosomes when the cells get poisoned. You want a source? I'll give you two. Read My Contagion Myth by Dr. Thomas Cowan, who to my knowledge is a psychologist or psychiatrist. Is that right?
Cowan. I made a video about him, but something about my videos is that I don't actually try and figure out the background of the people I debunk a lot of the time. I don't really care about the credentials. I just look at what they say.
Yeah. And then another one, What Really Makes You Ill by Dawn Lester. Now, if somebody were to want to establish that medically as the way that, the truth or how the reality of it, would you do it by publishing a book, or would you maybe do by publishing in a peer-reviewed journal like Nature?
You definitely, you definitely want to publish in a journal.
Nature's more physics. What's the one that's kind of the leading medical?
Nature is also Nature and Science cover all of science.
Yes, yeah. Is it? But all right. So is there one that's a little more medical biology? That's kind of the
Cell.
The top tier. Okay.
Cell is a top tier medical or scientific journal.
Yeah. So Dr. Thomas Cowan, maybe you want to go for that. Sorry. So then he goes, now let's get back to business. Enter Dr. Cameron Kyle Siddell. This frontline New York City doctor states from firsthand experience he's seen a lot of patients brought in and suffering from what seems like altitude sickness, a severe shortness of breath and lack of oxygen. He says they should be treated for hypoxia, and I think he miswrote it here, and not for ARDS, or acute respiratory distress syndrome. Now, could be true, could be not. I don't know if that's the case, but here's the deal. He goes on, they need oxygen, not pressure. He states that the apparent COVID-19 patients don't appear to be suffering from anything resembling viral pneumonia. Little did he realize that in his video, the effects of 5G on the human body at 60 gigahertz. So that's where my thing. It affects the oxygen molecule, causing the human body not to be able to absorb oxygen. So he's claiming that oxygen is modified so hemoglobin cannot bind to it. Which is more of a particle or high-end physics question than a biological question. Really, the thing that you're wondering about there that he's claiming is, does the 60 gigahertz photons, when absorbed by oxygen, does that change the chemical properties of oxygen? Which it does not. He doesn't provide any evidence. He just makes the claim. That's not, we'll come back to that. I'll come back to that. But the deal is maybe where he's talking about I don't know. I don't know if we need to, if what Dr. Cameron Kyle Siddell is saying really makes any difference at all. It's more about the exosomes. I'm sorry.
Yeah. I'm not sure who Dr. Cameron, whatever is either.
Back in April last year, he got a lot of videos on because he was, questioning how people were being treated, and if ventilators was right, or if maybe they should just get oxygen, like I guess like in the nose or something, the little deals. So
yeah, I kind of remember the kerfuffle about that, and people don't really go on respirators unless they're in really bad condition. So that's the only reason someone might be able to throw out a stat where like, oh, these people who all go on respirators, they died. Well, yeah, they were in really bad shape, and a respirator is risky. It's not good. It's not good to be in that state. But the alternative is just letting the person suffocate most of the time.
Yeah.
So,
and what causes that suffocation?
Well, so in the case of COVID, they're basically going into respiratory arrest. Their lungs are so damaged that they can't breathe properly, and so they're suffocating because COVID has damaged their lungs and likely allowed bacteria to come in and further damage the lungs.
Yeah, so that it's a pneumonia type bacterial infection on top of the other stuff.
Mm-hmm. And there is a phenomenon called COVID lung that people are starting to talk about now.
What's that?
Well, it's just pictures of COVID patients' lungs, and it's pretty dramatic. If I can bring up a picture of it.
We'll prepare for the presentation.
Let's see.
And then after, I think what we could do, we go after this on something interesting. Well, you're looking that up.
Yeah.
He does have he makes these claims of 5 different things that are the cause. And I will show that maybe after you talk about COVID lung.
Yeah, I don't have too much to say about it. Just
okay.
Just to show a picture here.
Let's see. And somebody's asking about talking about Kaufman's statement on virus isolation. And of course, that is kind of what you talked about already, but I think we can maybe go through the different things that confirmed the actual like studies that confirmed it.
Oh yeah, right. I could
I know
I'll go through that
you
in
have
a little
lot
bit.
Of those, so
here is a still image of normal lung, smoker's lung. So your last guest was a smoker. He seemed to be wanting to quit, but he was clearly okay with smoking. And that's a image of COVID lung.
So cloudy. The cloudiness is telling us things, right?
Cloudiness is bad. Pretty much all you would need to know here. That's not good. That's very not good. And it's something we can measure to see that a patient's lungs are damaged by this viral infection that we can also detect by other means.
Okay. So, all right. Let's go through the steps that you talked about. Do we have like the publications of these different things that have happened?
Oh yeah. let's see. So there are several. I think these 6 steps were pretty much satisfied with 2 main papers. If I could remember what they're called, I could find them real quick. I didn't prepare them. I should have prepared them.
It's all right. Let me while you do that, I'll
Yes.
read out. You don't really need to listen too much probably, but I'll read out what Goofball said here. The claims, and he is very verbose in his writing here. Here is the big thing here. He says COVID is an umbrella term for fakery, and all these different things that are being ascribed to it, which of course would be difficult since there is a genome for the virus.
Oh, yeah.
So he goes back to the Cameron Kyle Seidel claims, more about, I think he likes the correlation between the people that have COVID-19 and the installations of 5G, and the, you know, usage or the locations that have more air pollution, which of course you will find in more dense areas. So he found these correlations where lots of people live, there's air density or air pollution, and there's 5G, and there's large uses of other things like GMOs. And biofuels get burned more in areas of high population because there's lots more vehicles there. So yeah, so he says first air pollution, second GMOs, third biofuels, fourth vaccines. The residents of Wuhan were reportedly mass vaccinated before the outbreak. Of course, he has no like particular citation.
Vaccinated
for that.
to what?
Yeah, don't know what, but yes, people get vaccines. It's no surprise there. And then finally, he says EMF, which is Wi-Fi and 5G. The world has got out of the 5G 60 gigahertz frequency interferes with oxygen absorption, which it doesn't. It's just a coincidence that the NHY doc above is, yeah, the doctor is revealing that people now have symptoms of hypoxia or hypoxemia. So there it is. His list of claims. How are you doing on the
oh yeah, so one second here.
Some people have shown up from the Irish Demon stream, and they say a drunk leprechaun sent them. And they're asking, where's the other guy in the thumbnail? That would be Logan Mannix, who has received a link, who in fact challenged lots of people to debate. And you can find this one on his Facebook, which says, I will debate anyone on the subject, including medical experts, and you will lose.
And he doesn't show up, I guess.
It doesn't show. And this one, let's see, that's I want to find here it is. This is the one where he specifically challenged me or people in the chat. This is on your video there. He says, I'll debate anyone. I don't, whatever, if it's Fauci, Birx, I don't know who that is, Gates, at the same time. And then later he's like, I meant in the comment section. Like, I don't, does anybody think this type of challenge means in the comments section? Like, did he think that Fauci, Birx, or Gates would show up in the comment section of this video? I mean, I'm sure it would be excellent for Dr. Wilson for that to happen, but I don't think he really thought that Gates, Fauci, or Birx were going to show up in the comments section.
I doubt it. I doubt it. Yeah.
I'm pretty sure that this loudmouth do-nothing guy was meaning that he would debate live, but he doesn't have doesn't quite have it in him to debate live. So
yeah, interesting.
I found that the people that make these aggressive claims like that often are quite scared when you accept their challenge. They tend to run.
Mm-hmm. It always seems to be the case. Yeah.
Yeah.
But in any case, yeah. So I'll go ahead and share my screen here.
Okay.
Real quick. So this is a brief report in the New England Journal of Medicine, and this is, I believe, one of the first, if not the first, isolation of a mysterious pneumonia disease caused by a coronavirus in China, 2019. And so
it's a little bit off to the side in the view here. Other way.
Let me full screen it.
There we go.
That'll work. Is that good?
It's moving it around. There. Well, it kind of pops around. There. Well, it's all right. I think we can see the title, and people, if they wanted, they can look it up. I think maybe what I'll do is
you mean like it's a little. You want this to be centered?
Yeah, it's okay.
Oh, that's just the way it's laid out.
Okay, a novel coronavirus from wait, what did that say?
Oh, sorry. From patients with pneumonia in China.
There you go.
So they didn't know. People were getting sick, and they were trying to figure out what's causing them to get sick. Obviously, the doctors probably suspected it was maybe influenza, maybe some other known thing causing the illness. But what likely happened was they did those tests and they came back negative. So they could not find the virus or the pathogen that was causing this illness. So they looked at the samples more closely, and they found a coronavirus. And they realized when they sequenced its genome that it was a new coronavirus, that this genome of this coronavirus was unlike anything that we had seen before. So you can read through their methods of how they collected bronchial lavage. So this is lung samples from these patients who had pneumonia, and then they extracted the nucleic acids from those samples. So this is what we would call an isolate. So they brought the sample out of the patient, and instead of doing other things with the real virus, they just extracted the nucleic acid to sequence it, because they were like, what is this thing? We don't know what kind of virus this is. So once they sequenced it, then they figured out that it was brand new. They also had images of these patients' lungs that were pretty dramatic. Patients were very sick. And then they imaged these samples under an electron microscope. And what they saw were particles that had a characteristic coronavirus shape to them.
Yeah, you can see, it might not come through in the stream, but if people get the actual study, you can see in the image the little nubs off to the side.
Yeah, so where coronavirus gets its name is it has these spike proteins that look like little lollipops sticking out from them. And it gives it a kind of, well, when the people first saw it, they thought it looked like a crown around the virus. And so they call it coronavirus. So this is characteristic of coronaviruses. Now, something I go over in the video that this guy's commenting on is that you can't identify what a virus is with just scanning electron microscopy alone. Here which is what the guy in the video that I was debunking claimed to have used. Here they're using transmission electron microscopy, which is higher power, lets you see smaller things, essentially. But even so, you see this and you say, that looks like a coronavirus. But what allows them to actually say that it is indeed a new coronavirus is the fact that they had the genomic sequence along with it. So they were able to extract a sample from this patient. They were able to isolate the virus, sequence it, image it, and then other studies.
Now that was published in 2019.
Oh yeah, this was. Let's see. Well, they did the work in 2019. It looks like it was published in.
Oh, February of 2020. Okay.
So, article was published on January 24th, 2020, okay, and updated on 29th.
So they isolated it, they sequenced it. So when like Logan Mannix says that the Chinese version of the CDC did not, I don't know what he's saying, did not isolate it or did not something. This right here shows that it has been sequenced and isolated.
Oh, 100%. Yeah. Okay. And let's see. Something I like to point out with these. It might be awkward for me to find it on stream, but somewhere in the paper they will talk about, here. They name the isolate. So this is the nomenclature that was used to name the isolate. Mm-hmm. I can take this, right? I can go to PubMed. I can, let's actually go to here. I can make sure I'm searching for gene, or better yet, let's go to nucleotides. I can paste that isolate name in the search bar. Oh, items found. Do I have to go to, awkward. Genome. Okay.
There's a space on the end. I don't know if that changes it.
So the point is you can find these genomes banked in particular places. And so, well, actually, I have the website pulled up already. I should just go there. This is a site anyone can use. It's called NextStrain.
Ooh, okay.
So this is all of the isolates, all of the coronavirus isolates, SARS-CoV-2 isolates that have been gathered since it emerged 2019.
So this, any strains that have different genomes?
So these are isolates. Some of them have identical genomes.
Okay.
But a lot of them are variants, which just means that it's an isolate that has some nucleotides in its genome that differ at least in one spot from a reference isolate.
So this has been isolated many times.
Many times.
So anybody saying it's not been isolated, every one of these dots is an isolation. Is that right?
Yes.
Yeah. So,
and you can go, so here's a database here that is linked in, so you can click on any little point in this dot plot in this tree and find the author of the paper who that published the isolate, and you can find where it was isolated, and it has a little reference here. If you click on that, it takes you to a database where you can actually see the SARS-CoV-2 isolate genomes.
So these the talk about these newer strains that are like the UK or Brazil strain, are they they're also in there?
Oh yeah. They would be listed. You would have to yeah,
there's a lot. So you have to find them. So
yeah.
So they're asking what's the link for what we're looking at. And he's gone through a few different things, right? So PubMed is one of them.
Oh, the link. Yeah. So I can actually, let's see. I'll paste this link in the Zoom chat and you can give it to your audience.
Yeah. Okay. And what I will do is I will put on my website a collection of these different documents. So if anybody's like, well, what about where can I go to find information on whether or not it was isolated? It's all out there, but for people that aren't in the industry, it's difficult to find, right? We don't know.
Yeah, I mean, it's pretty simple, but you have to know where to go.
Yeah.
Yeah.
So I have one in particular that I use that's or two. One that shows that actually has like the genome, all the letters listed in the genome, right? And it's been isolated. And then the other one that specifically says it's satisfies Koch's postulate for viral diseases.
So here's just real quick, here's a reference genome to a SARS-CoV-2 isolate on PubMed. I'll put that in the link in the Zoom chat as well. And if you go down, you can see this is the amino acid sequence. So this is the genome translated into protein. And then if you go way down, this is the genome.
Yeah.
The A's, T's, C's, and G's.
That looks, I've seen them before.
Yeah. You can download this yourself. You can use a ton of programs out there to like ask certain questions about it, compare it to other genomes, all that stuff.
Yeah.
Okay. But it's all freely available to see, which is nice.
So we got Cinnamon Control asks, I see lots of claims of, but sequences are just computer generated. What about that?
They are not. So you have to actually have material to sequence. You have to have physical nucleotides that you have extracted from a sample. And there are several ways to then determine the sequence. And the cool way that's, I think, easiest to explain that the original isolate actually used, in part at least, they used a couple of different techniques, but one of them they used, it's called nanopore sequencing. So what you have is a silicon disk with a little nano-sized pore that is only wide enough for a strand of DNA or RNA to fit through at a time. And you have a buffer solution on one side of the pore and on the other side, and you can create a current that will create an ionic flow through the pore.
Okay.
And you can measure the current of the flow through that pore. And anything that comes through that pore and physically blocks that current is going to change that measurement.
Oh, okay.
And so
you know when it comes through.
Yeah. So as the strand starts coming through, you can see that. And now the bases, right? You've all seen the double helix model of DNA.
Yeah, yeah.
RNA is similarly shaped. It's usually single-stranded. But in this case, no matter what, it's a single strand coming through the pore. And those bases, the things that make up the rungs of the twisted ladder, the A's, T's, C's, and G's, those are all different sizes. So an A is a different size than a T, is different size than G and C. So as they come through the pore, they're going to block a different amount of current. And you can actually measure what the sequence is as it flows through that pore. That's a cool way to sequence nucleic acids. And there are several ways to do it, but it's not computer generated. You have to physically measure. Of course, it's measured by a computer, but how else are you going to measure a current?
Yeah, it's people that they're just deniers, and of course we see that all the time with flat earthers and stuff like that. That they don't understand, they don't know how it works, they don't work in the industry. They tend to be almost always, quite uneducated, and they're just like, I don't want to believe it, so I'm gonna just say that it's just computer because I don't want it to be true. So.
Right. So we have the we have the isolate, right? So that's good. We have that satisfied for the Rivers modified Koch's postulates.
Yep.
We have the isolate, we have the cultivation and host cells. These samples are usually filtered before they are put into culture. So, you know, that part of the goal of putting these samples into a cell culture is to figure out whether or not it's a virus or bacteria. And they can figure out whether or not it's a bacteria pretty quickly because you can visibly see the bacteria starting to grow and outcompete the cells in the dish. And you don't want that if you're trying to figure out whether or not it's a virus.
It messes up your sample.
Exactly.
Yeah.
So you would filter it first before you put it into cell culture. So all those three things are satisfied just from isolating the virus and cultivating it in host cells. So then a production of comparable disease in a host species or original or similar species, and then re-isolation of the virus and detection of an immune response. So let me download the PDF of this and share that. This is a paper published in Nature where it was a mouse model that was adapted to study COVID-19, or how SARS-CoV-2 infects its host. And so SARS-CoV-2, we knew enough about coronaviruses when SARS-CoV-2 was discovered to know that coronaviruses, like SARS-CoV-2, usually try to get into cells by hijacking a protein that our cells naturally produce. And this protein is called ACE2, or angiotensin converting enzyme 2. You don't have to know what it does, just know that it's a protein that sticks out on the cell surface. The virus can recognize that and use it to get into host cells. So in order to study this in an animal model, because again, studying viruses in humans is unethical if you're infecting them on purpose, unless you somehow get permission to do that and everyone knows what they're doing. You want to use an animal model, and not every virus can infect every animal. Yeah. So these researchers made recombinant mice. They engineered mice to express ACE2. That's the only thing they changed. They just made it so that these mice can express this human protein. Oh. And when they did that, and then they exposed the SARS-CoV-2 virus to these mice, then they observed a bunch of effects. And so I'd have to quickly remember what all these panels actually mean. But I think it suffices to say that they have unmodified mouse and modified mouse, and they infect both. They expose both mice to SARS-CoV-2.
So a test and control, as a good experiment has.
Mm-hmm. Yeah, imagine that. And they see that the mice that don't have ACE2 expressed are not infected. And they see that the mice that do have ACE2 expressed do get infected. And you can see, so they end up measuring very specific damage happening in the mice tissue because of the virus. And they measure a whole bunch of things. They measure aspects of the disease. They look specifically at lungs. They look at more tissue markers. And so
if I can, they're measuring multiple different things. Each of these different things potentially is an independent variable in the kind of strict dependent independent variable thing. Or sorry, these are the dependent variables that they're seeing this change in, right? Yeah.
Right.
So multiple dependent variables.
Mm-hmm.
Yeah.
Yeah. And here actually this nucleocapsid panel, they're actually staining for the virus. So that brown signal, that's virus. So they're physically detecting virus in these damaged
yeah.
tissues.
Yeah.
So
nice.
They're able to demonstrate all of these things. And of course, we know that we can re-isolate a virus from infected people or infected animals.
Yeah. So you know what you put in, and you then get out the same thing.
Mm-hmm.
Yeah.
Yep. And so this is all satisfying Rivers modified Koch's postulates. You start with a sample, you figure out that there's this pathogen in it that you think is causing disease. You want to test to see if it's really causing the disease that you see in all these patients. So you put it in an animal, it causes an illness that is similar to what you saw in humans because it damages lungs. And then you can see that the virus is in fact there. And that is causing, damage to the tissue. You can measure mouse antibodies that are made against the SARS-CoV-2 virus. So it's really straightforward for labs that have those capabilities to do all these things. And they've been done by labs all over the world. So, I mean, really, if this is a conspiracy, then the whole world has to be involved.
Yeah.
So,
yeah. That's significant. Yeah. They talk about the certain president couldn't keep two people quiet. It's tough to keep the larger it gets. Interesting. Yeah. So, all right. So I think that's great. And I'll work with you, and I'll get maybe a rundown of that and have it on my website. It will probably be mc2.net/cv19.
Mm-hmm.
I'll work on that in the next few days. Now, another question or claim that they talk about is the PCR test. Can you speak to that?
Yes. I would love to talk about that. But one thing I want to move on for, I want to do before we move on, just because it's relevant. So you might hear some people complaining, particularly Andrew Kaufman, complaining that people have isolated the virus, but no one's purified it. Well, isolation and purification are two different things, because when you pull up a little bit of liquid from a culture of cells that has been infected by the virus, that could have cellular debris and cellular excretions. It could. So it's technically not purified, but purifying it is rather simple. So what these guys did to purify SARS-CoV-2 viruses, virions, is to use centrifugation, sucrose centrifugation specifically. So they make a density of sucrose, a density gradient of sucrose. And sucrose is just it's a sugar that if you dissolve it in water, the water becomes really thick. And you make layers such that the top layer of sucrose is less dense than the bottom layers. And then you can lay a biological sample on top of that sucrose and then spin it at ridiculously high speeds in a vacuum. And as it spins, the heavier molecules are going to get pushed down into the sucrose until they reach a point where they're in equilibrium with the density and the centrifugal force, and they'll stay there in a specific band in that sucrose.
So it's a density tower in a sucrose gradient.
Mm-hmm.
That's genius.
Yeah, I've done this experiment several times, and it's pretty fun. Once you do that, you can have a whole little tiny section of sucrose that is filled with just virus, because they're all going to be the same size, same weight. And that's what they did. And they put these samples into a cryo electron microscope, which is a type of transmission electron microscope. And they got these lovely high resolution pictures. Sorry about that. And you can see much more clearly than before, I hope.
It's the nubs again.
These little lollipops sticking off of these viral particles. And Andrew Kaufman might say, oh, well, that looks like an exosome. How do you know that's a virus? Well, the nice thing about cryo-electron microscopy is that it lets you view things in atomic resolution. So you can literally see the atoms that make up the spike protein that is the little nub that sticks out from the virus. So by imaging, hundreds of thousands of viral particles, they get enough data to build a high resolution model of what they see. And this is an example of what you would see. And if you're interested in playing with this or looking at this structure in more depth, you can look at what the authors name it in the paper. So I remember what the code is. So every cryo-electron microscopy structure that people publish, they have to deposit it in a public bank. And it gets a little PDB ID, Protein Data Bank ID.
Oh, yeah.
So I can go to Protein Data Bank. And there it is, the structure of the SARS-CoV-2 spike protein.
Wow.
And if I want to play with it more, I can have this little this program called Pymol. And using this program, I can.
Do you write in Python?
I no. This is someone else's program that this is a free.
I love
it might not be free actually, but it's a.
I like Python, so I write software.
But I can tell it to go fetch a protein data bank name.
Oh,
and when I do that,
very nice.
Then it'll go fetch it, and you can see it's the spike protein.
Oh yeah.
I can tell it to then show me the sequence. So this is the amino acid sequence. I can select them and see where they are in the structure. I can turn it around. I can zoom in on it. And when I look at the structure and see the sequence of amino acids, I can go back to the genetic code and see that it matches. I can see that the nucleotides that code for specific amino acids should make these specific amino acids, and that's what we see. So really, really amazing work that people like Andrew Kaufman are kind of just ignoring, and I think it's his loss. It's kind of sad.
That is, I love that.
This is what I played with all the time in my graduate work. These kinds of structures.
Wow. And so have multiple places done the same analysis of these spike proteins and gotten the same results?
I believe so. Yeah. I think offhand there's one other paper that did it. I imagine there are several more.
Yeah, you could look up, and so somebody curious could because you want repeatability.
Oh yeah,
in science, and if a separate lab does the same thing and gets the same results, then that's a strong confirmation, and the more you get, the better the confirmation.
Yeah, so here's this is again the protein data bank. I just type in SARS-CoV-2 viral protein, and I get lots of hits. 3 hits to be specific.
So now these are different. Oh, because every time you sequence it, you get a different name to the database.
Oh, so this is not these are just different structures. So sequence is different from structure.
Okay.
This is physically looking at the actual protein that sticks out of the viral envelope, and visualizing it in 3 dimensions rather than sequencing the genome.
I see.
But since it's a protein, it's dynamic. So it wiggles around, it can change shape.
Okay, okay.
And this spike protein in particular, it has at least 2 different conformations. Could be wrong on that, but
okay.
At least 2 different conformations that it uses to recognize host ACE2 and then get into the cell. So it has to like bind, change shape, and then enter the cell. And so that's part of what you might see here. So for example, this is closed conformation. This is open conformation. This is another open conformation. And it's all by the same researchers, these results. But if I, look hard enough, here's another spike protein structure from a different lab, doing the same technique, getting a similar structure.
Yeah. Okay. And so now this particular, that when they say spike protein, this is what the antigens that fight it in our body are binding to.
So yeah, it makes sense, right? So it's because the spike protein is on the outside of the virus, that's what your body's going to see. So it makes sense to, when you make a vaccine, have it target that spike protein. And so that's what the mRNA vaccines, they have the spike protein coded in the mRNA.
So does it actually have like a replica of that protein in it, or is the opposite or something? How does it train?
No. So it's the way the vaccine works, the mRNA vaccine works, is it delivers the RNA code. So in order to get a protein, you have to have, at least normally in humans, you have DNA, which is like the library. That's how most people describe it in school. It's like the library where you keep all the information. And then you have RNA, which is like somebody goes to the library, they make a copy of a book, they take it with them. And then you have the protein, which is, if the person wanted to use that book to make something, to build something. So there are different mechanisms in our cells that read DNA, and they'll make a copy of it in the form of RNA. And RNA is much less stable than DNA, so it gets sent around to the cell to where it needs to go. And then once it's ready, the mRNA will get read by a different set of machinery, and that machinery will produce protein. And one mRNA molecule will produce multiple proteins until it decays over the course of hours, sometimes days. And then you have your protein. So what the vaccine does is it delivers the mRNA to your cells. And the mRNA codes for only the spike protein. And your cells will read it, just like it reads all the mRNA that your body is constantly making. And it'll make some spike protein. And once it makes that spike protein, then your body starts recognizing that as foreign. And thankfully, since there's no virus behind the spike protein, it doesn't make you sick. So that's an advantage of vaccines.
Yeah, and that's different than like the attenuated virus type, where it is.
Yes.
It's there. It's supposed to not infect you, but it is there.
Mm-hmm. Yeah, it's separate from. It's a different kind of vaccine.
Yeah. Awesome.
It kind of skips the middleman.
And so that's a potential to be better because you're not introducing the actual virus into you.
Yeah, it has advantages. We don't know of many disadvantages yet, but it definitely has a ton of advantages, including one thing that anti-vaxxers always harp on is, they say they have a problem with how viruses for vaccines are grown. So they have to be grown in cells.
Yep. So something alive
Mhm.
had to be there to grow in.
Yeah. So some anti-vaxxers have problems that viruses are grown in cells and then used for vaccines. But this mRNA vaccine, you don't need any cells. You can just make the mRNA. There are molecular techniques where you can synthetically make strings of nucleic acids. And that's what's done here. So it's really cool in a lot of ways. But I believe I think you had a question about PCR.
So yeah, so this Logan guy, and it's common, they say that the PCR test, something, the guy who invented it, something, can't use it for that. So what did that guy actually say? What did he mean? And what can the PCR test do or not do?
So it's actually really interesting. I'm kind of spoiling a video I have planned in the near future.
Be sure to subscribe right now, people. I'm going to put his link in again. The chat.
So the story goes that this guy Kary Mullis is the inventor, credited with inventing PCR. Obviously, it wasn't just him because science doesn't happen in a vacuum, but he is credited with it. He won a Nobel Prize for inventing PCR. And he's quoted by several COVID deniers saying that you can't use PCR to diagnose viral infections. And Kary Mullis goes on to say in this quote that, you can't use it for diagnostics because it's not going to, he says something like it's not going to detect specific pieces of nucleic acid, which is not true. PCR can be very, very specific in what it detects.
Okay.
So he did say something like that. But what's really interesting that people don't end up really looking at is, the original patent for PCR. I'll bring that up real quick, which has Kary Mullis's name on it. So here's the patent, Kary B. Mullis, right there, 1986, long time ago. It's describing PCR, process for amplifying, detecting, or cloning nucleic acid sequences. This is what PCR does. If you search through the, if I just do a little search for diagnose, excuse me. There's a portion of this. There's a portion of this patent, right? There it is. It says various infectious diseases can be diagnosed by the presence in clinical samples of specific DNA sequences characteristic of the causative microorganism. These include bacteria, such as Salmonella, Chlamydia, and Neisseria, viruses, such as hepatitis viruses, and parasites, such as Plasmodium. So
that's broad, yeah. So, but now he says DNA, but then also viruses which don't use DNA?
Same concept. So PCR can detect either DNA or RNA. It doesn't make a difference when it comes to how PCR works. So it's true that he said this. So why did he say that? Why did he say something that contradicts what he wrote in the original patent? Well, that video of him talking about that is he was being asked a question about HIV. And Kary Mullis was an AIDS denier. He did not think that HIV causes AIDS. So in that video, he is saying that you can't use PCR to diagnose HIV because he is denying basically that HIV causes AIDS.
I see.
Which, if you're going to defend that stance, I think you've got to take a couple extra leaps.
Yeah, I think you also have to be with him on his stance on HIV and AIDS. Yeah.
Right. So he was a kind of eccentric guy. You know, he would also talk pretty seriously about his experiences with LSD. So, I mean, that's not objectively bad, but you know, he would seriously talk about experiences where he spoke with glowing green space raccoons. He was a unique guy. So, you know, it's like COVID deniers have this thing where, you know, they trust authority, but they don't. You know, they say, look at this guy. He won a Nobel Prize. He invented PCR and he says this. But then they don't trust someone like Anthony Fauci, who has been in his position for decades as an infectious disease expert. They're picking and choosing who to believe, and
it's cherry picking is common amongst the science deniers. Yeah.
And then they also tend to appeal to authority in that case, where they say he won a Nobel Prize, so he's saying this, you know, that must be true. But in science, you know, we don't appeal to authority. There are several Nobel Prize winners who have said things that are completely outlandish. Linus Pauling was one he won 2 Nobel Prizes, and then later in life said that vitamin C cures everything. There were, of course, Watson and Crick, who won a Nobel Prize for discovering the structure of DNA, which Rosalind Franklin really helped them with a lot, but she wasn't credited. But they were deeply racist. They held deeply eugenic views that were based on, you know, inheritance and genes. So yes, they won a Nobel Prize for that, but are you going to take seriously their ideas about eugenics and race superiority and misogyny seriously? I don't think you need to.
That is the nice thing about science, that the reality of the world, of the universe, is independent of the opinions of the people that discovered it.
Mm-hmm. Yeah, exactly. So the whole story with Kary Mullis is just, you know, the guy kind of fell into some weird stuff later in his life, and that's why he went on to say that PCR can't be used to diagnose, when in reality it was designed to do exactly that, and it said so in the patent. That's what we use it for all the time. And when you understand the way it works, it's really, you can't really argue with the idea that it can be used to diagnose an infection, not disease, an infection.
Okay.
So, I don't know if you want me to go over how it works or.
No, I think that covers specifically Logan's thing.
Got it.
And we've had some comments from the audience here. I think it'd be good to go over some of them. People like you a lot and want you back and want more conversations like this. So I'll read some of them. We've got PhD Tony. PhD Tony is Australian. He's a PhD. He is Daniel Pratt's twin brother.
Oh, cool.
If you you know Daniel Pratt. Yep. He says, thank you, MC2, and always great to hear from people who know what they're talking about. He's talking about you. We've got let me change this here to we've got Irish Demon says Bon Jovi, as the French would say. I think bonjour. Bonjour.
That sounds right.
Sorry, Ed, he's Irish. You can't understand half of what he says. Anyway, John Eagle says, should I order my zombie apocalypse samurai sword today or wait?
Oh boy.
I think we're on the downside of that, the other side of that peak there. I hope so. Wait. We'll see.
Hopefully we are. I'm hopeful.
Rodent, no last name, says, this is awesome. More, please. So there you go. Rodent, you can get all of the Dr. Wilson you want by going to the link I just put in the chat there and is in the description. He has a good number of videos, and the this is just a fun side effect of his videos. If you start with the old ones and then move through the newer ones, you will see his hair get more and more awesome over time.
You can also see my first YouTube video, which was really bad.
You know, they mostly are. I tell people who are they're like, I want to start something. Just start something.
Oh yeah.
Doesn't matter. Your first video you can take down later, or you can leave it up. It doesn't matter, and leave it up for comedy if you want. So, and one more, and I think this is for a topic for a different show. Maybe if you'd want to come back. If it's Secretan Bull asks, if it's not too far off topic, can Dr. Wilson share his thoughts on the future of CRISPR? Can you do that in 1 minute, and then maybe have a different topic a different time?
Yeah. Well, the future of CRISPR is massive potential. That's the probably fastest way I could sum it up. I mean, CRISPR, if anyone's seen the movie Gattaca, I don't know if you know what that is. I have not seen it. Gattaca is an old movie it's a sci-fi movie about like, what would it be like if we could genetically engineer human babies? Like it's a kind of dystopian sci-fi thing. That was fueled by this discovery of recombinant DNA technology. Because people thought, oh, we're going to be able to edit genomes now, and it's crazy. But recombinant DNA technology, if anyone might know what that is the capabilities of that were nowhere near that of actually editing humans. With CRISPR, that possibility becomes more feasible. And of course, there are a ton of ethical questions there, but what gets me excited is the idea that we would be able to treat genetic disease. So terrible diseases like cystic fibrosis, where, people essentially live their lives knowing they're going to die young.
Yeah. And I knew somebody that died from it is horrible. Yeah.
Yeah. I mean, we're not there. We're not there yet, but the idea that we could cure someone of that is feasible because of CRISPR. There's a ton of work to do. There's a lot of nuance and little things that need to be figured out in order to actually make that possible. But it's feasible with CRISPR, and that to me is just huge potential.
Yeah, that is cool. Okay. Let me, if you don't mind, I will do my part of the claims from, and I'll be a little quicker on this. Let's see. He made a lot of claims. And oh, did I find it? I think I dropped it in here. So he talks about 5G being the cause. So he says, and let me, be specific here. He actually brings up Mark Steele. So this long rant he goes on here, he talks about the video that Mark Steele did a while ago with that lamppost. And in the lamppost, Mark Steele pulls out a this thing, an antenna from the lamppost. I have one of these antennas right here. It's a really basic antenna, which I'll go over in a second. But to read it here, he says there's a thin cable from this unit that goes to the LED streetlights, but there's a bigger cable, a lot thicker, that connects to a number of 450 volt capacitors in the sealed unit that connects back to the scanner radar that's also connected to 450 volt capacitor as well, which I'll show you right here. This is what he's talking about. These little things here are 450 volt capacitors. That's just the maximum voltage that they can handle. It doesn't actually mean anything. Anybody that knows electronics knows that that's just a normal capacitor. It's not a big deal. You can't actually store much energy in there because of the capacitance of it is also very low. So the amount of energy stored in there is tiny. Anyway, you don't need that kind of power, which he didn't figure out how much power was in there. In that kind of ballast, LED does not need large amounts of energy, and there aren't large amounts of energy. A 450 volt capacitor with just a couple nanofarads or microfarads. Do the math. You can do it yourself, Logan. There's a capacitor energy calculator online. You don't even have to know the math. Type in the capacitance and the voltage, and you'll find out how much energy. It's tiny. So I do have one of these right here. It's the same as in there. Now, this is an omnidirectional antenna. I know that because it has a single conductor going up the middle there. On the back, it has nothing, not covered with a grounding material. It's not grounded. It can radiate in all directions. It is a 1/4 wavelength antenna, which is the quarter wavelength of the 869 megahertz that it's designed to work at. In fact, and I don't have it right now, but I'll put it in the description. There is, I have 2 different articles discussing how to build design a 868 or 915 megahertz quarter wave antenna on FR4 substrate, which is what this is. It's just a fiberglass, mostly substrate. And the dimensions of that particular one are these dimensions exactly to the millimeter. So this is an omnidirectional antenna. The documentation for how to do it is out there. On this board also is a 100 milliwatt software-defined radio. It does not have a heatsink on it. And if it doesn't have a heatsink on it, it cannot run at full power for very long. It would burn itself out. It's supposed to be tuned for 25 milliwatts. And the FCC documentation for this, the 915 megahertz version, which I can also share with you, Logan, should you care, I'll put in the description, specifically says that they measured it and it has the expected EIRP for this particular 25 milliwatt usage. And finally, my friend Lee from Lee's Lab tested this exact same unit, hooked it up to his own software-defined radio, and also he got the controller to turn this on as well, and was able, and then tested it in 360 degrees. It is an omnidirectional antenna, and then finally there is not enough power in there. Oh, it can't run at full power all the time because it doesn't have a heatsink. It is regulated by law to run at 1% duty cycle, which means it can only transmit once every 1 second out of 100 or whatever, as long as over time the average is no more than 1%, which matches the lack of a heatsink on there. And then the final thing, and this is the really important thing that he talks about a lot, is 60 gigahertz 5G. Now, when I first heard about 5G, I saw a lot of things talking about 60 gigahertz, but then I looked into it and I found one thing that was very interesting. 5G does not use 60 gigahertz. Not anywhere, not ever, not at all. And how do I know this? Well, I know this because first, it's been documented. So I have right here on my website, mctune.net/5G. You can look at the second link down is the cell phone frequency allocation. And then it has the different spectrum allocation for different countries. In the United States, 5G has been allocated at 700 megahertz to under 5 gigahertz. And you can just go through right here, Logan, or anybody curious. You can see what the FCC has said is legal. If they go outside of this, they can get in trouble. If you think they are going outside of this, get yourself a spectrum analyzer and tell on them, please. I don't want them breaking the law. Go telling them hefty fines if they're doing that. At the bottom here, it does talk about the higher frequency, the frequency range 2, which is the high frequency in the gigahertz range, 26.5 to 29.5, 24 to 27, 37 to 40. None of these are 60 gigahertz. Excuse me. So that is the documentation from the United States. There's the UK, Australia, Canada, Brazil, others. You can look it up yourself. If you're curious about whether or not 5G uses 60 gigahertz, look up what's legal. Now, if it is not allowed, now, you can look at the cell phone carriers as well, or the cell phone manufacturers. Apple lists all of the frequencies that their phones use. So do the other companies. You will find that they do not list 60 gigahertz as one of the cell phone carrier frequencies that they use. So the cell phone companies don't say they use it. The regulatory agencies say they don't use it. The handset manufacturers say that they don't use it. So unless you have evidence that they use 60 gigahertz to broadcast cell phone, they don't. Now, here's the other thing. Even if they did use 60 gigahertz, because it is used somewhere, some places, 802.11.AD and 802.11.AY both do use 60 gigahertz. It's very limited. The hardware is very expensive and it doesn't work well. So the market for it is rather small. Does it actually do to oxygen what they claim it does? The claim is, and Dr. Wilson, this I think you'll find funny when I covered it briefly, but they say that the oxygen molecule, which is actually two of them, two atoms of oxygen together, bound together in dioxygen, and it's specifically in triplet state. And the electrons in the shell, the outer electrons are paired. Triplet state is the normal state in air, and that's what our bodies use. Singlet state is another form of it. It has a different chemical interaction property. It is not what we see, and 60 gigahertz doesn't have the energy to convert it to singlet state. You can do the math to find out exactly using the Einstein-Planck formula to find out how much energy is in a 60 gigahertz photon. It's not enough to move it to singlet state. All it does is it changes the spin properties, which I'm sure you've seen, of the two electrons there. Because of the Pauli exclusion principle, there's only certain places that they can be. But when you change the spin, it still chemically responds the same way because it's a mirror image. And so the oxygen would just bind the other direction if it does have a magnetic issue. So it doesn't. Sorry. You can look up, Logan or anybody, the experiments by Van Vleck in the 1940s. Yes, we've known about this for a long time. The energy levels that move electrons between different hyperfine states, or he just calls them superfine, I think, in the paper. And again, I will put that in the description. So there you go, Logan. If you think that 60 gigahertz is in use, you would need to find some evidence of that. And if you thought that 60 gigahertz changed oxygen's properties, you would need to find evidence of that, which I know you don't. In fact, I've asked 5 of the people who are the biggest claimants that 60 gigahertz makes that distinction or that change to oxygen in a video. No responses from them. I've contacted every one of them, except for Kate Shimarani, who's a nurse. She doesn't have a clue on these things. None of them have the evidence to support it. So there you go.
Huh. Wow. Yeah, I mean, even if that were true, even if 60 gigahertz somehow did affect oxygen and it hurt us, why is it only some people who are getting sick? Why wouldn't everybody be experiencing?
Why wouldn't frontline Dr. Cameron Kyle Siddell have that very same problem? If he's in the same vicinity as all of these people that are having it, he should have the same symptoms.
Yeah. Everybody in an area should be getting sick, and that's not what happens.
And hypoxia, as soon as you get oxygen, you get better, right?
Yeah.
Well, if 60 gigahertz is causing hypoxia, all you would need to do is not be near the 60 gigahertz. Then you'd get better. So that must be in high intensity in the hospitals where these people are on ventilators, supposedly causing it. Yet the nurses, the doctors, they're not having the same symptoms. It doesn't match. Correlation is not causation. But when there's a lack of correlation, there you have a problem. You can certainly make a case that there is no causation if there is no correlation.
Right. Yeah.
So,
if you don't have correlation to start with, you're not going to get to causation.
Yeah.
So,
I mean, it is necessary to find that correlation in an experiment, and if you have isolated the other things out, then you have some confidence that that correlation is the cause. But there's still something else that could be the cause. You never absolutely know. So
yeah, and I know that Dr. Thomas Cohen, however you say his last name, he writes about this, and he thinks that all disease is caused by electromagnetic phenomena. I mean,
a lack of correlation again.
Yeah, we know that flu viruses and polio and smallpox have been around for centuries, if not millennia. And where do those come from? where was the electromagnetic radio waves back then? Back in ancient Egypt, where first cases of what we think were polio were recorded.
Yeah. And why, if polio was caused by EMF, why is it gone?
Yeah.
Apparently not the vaccine, apparently magic or something. I don't know.
Yeah. I don't know. It's it'll be interesting to get someone like sorry, I can't remember his name for the life of me. What was his name? The person who was supposed to be here?
Logan,
yeah. It'd be interesting to get someone like him on here to, maybe see how they respond to all of this in real time.
Well, I have had a couple different people that think 5G is doing whatever on here, and they typically they have. I was very surprised because it's not coming from a flat earthers often are like, well, the Bible says this. There isn't a fundamental, like, underlying religious aspect to thinking that 5G is bad, but they have the exact same religious type responses to it. You show them science, you show them evidence, you show them how this was done, you ask for evidence supporting their point. They do the same thing. They're like, change the topic, something else, or find some little thin thread of nothingness that they're like, I connected these two dots, and they drew a portrait between these two dots.
Yeah,
Ooh,
a dog shaking himself off.
Oh.
Yeah, I'm sure that some psychologists would have really interesting things to say about it, but to me, it's interesting the way some people latch onto these ideas.
Yeah.
And I used to be a conspiracy theorist, so I kind of understand that mindset. But
what was your kind of something top one of yours you thought?
9/11 was the big one for me.
It's common, yeah.
I bought that hook, line, and sinker before I started pursuing biology as a career.
Anything else?
Oh, I mean, Ancient Aliens was really getting me. For a while, I believed in some of the like fake cancer cures and fluoride ideas.
Oh, yeah.
Yeah, I mean,
they bundle up, right? And when you get to some of the ones that are in these more extreme places, they just have all of them, right? Because something clicked in these flat earthers. They cannot reject any conspiracy theory, right?
Yeah.
If you're like, just go down the list, they all, with a couple exceptions, take them all.
Yeah. So I mean, it's really tough to get out of that mindset.
Yeah.
It takes like
Well, they didn't get there with logic, so you're not going to get them out with logic a lot of the time. So sometimes they do. Maybe for you that was it, but
yeah, I mean, for me it was. I mean, I used to spend hours in comment sections just going at it with people, and it was so stupid. But you know, in part, the people who had the patience to keep responding to me, that persistence, I think that played a role in me eventually like saying, you know what, this just isn't right. This isn't making any sense. And then I just clawed my way out, got into science, and understood more and more why all of those views I used to have were nothing. There was nothing to them.
yeah. Well, good. I think we covered. Maybe there's a couple things that because this Logan has like all of them.
I'm sure he has tons more to say that we can spend hours going over.
But Logan, yes, you are an egg-laying poultry with feathers. That's what you are, Logan. These big words of "I will debate anybody," you certainly won't. So not very impressed with him. And he's former military. I don't, you know, the military people I know, they don't run away from their own challenges. So
yeah. And Logan, you know, I've got a family. I'm not going to spend too much time in comment sections arguing with people. So if you want to talk, come on here.
Yeah.
We'll talk.
All right. Well, that is it here. Thank you so much for coming on, Dr. Wilson. Maybe we can come get another topic and just talk about something and not even concern ourselves with some goofball.
MC Toon, it was my pleasure. I'm honored to be here. And oh God, okay, my dog is coming in to say goodbye. This is Toby. He's yeah. But seriously, just I don't know how to say I appreciate you and your audience enough. So I really, really appreciate being here and being someone you guys listen to. So
yeah, well, excellent. I'm honored for you to be on here. So I put your link in this description again. Go sub to Dr. Wilson, and his dog will be happy.
Yes, you will.
All right, thanks everybody. I am planning to do a St. Patrick's Day something short to celebrate, and I've invited Irish Demon, our Irish Leprechaun, today to join. We got one more super chat. Tech J for £5 says, I really enjoy ancient alien conspiracy theory. It's a lot of fun to think about. It is absolutely fun.
That's
to think
the thing.
about.
It's like, wow, that would be so cool if it were true. But then, you know, you really get into it and it's like, it's not.
Tenuous links. So, all right, everybody, we'll see you later. Bye.
All right.